Hello everyone. Children, we have already started with chapter number 5, that is Principles of Inheritance and Variations, in which we have started with the introduction of the chapter. We have learned about Mendel's law of inheritance, about the history of Mendel, Mendel's experiments, Mendel's experimental approach. Then we have studied about Mendel's experimental material, Mendel's experimental procedure, Mendel's observation on experiments, and Mendel's inferences. Now today we will start with the new topic, that is Punnett square. The production of gametes by the parents, the formation of zygotes, the F1 and F2 generations can be explained by a diagram called Punnett square. It is a graphical representation to calculate the probability of all possible genotypes of offsprings in a genetic cross. The 1 by 4 is to 1 by 2 is to 1 by 4 ratio of capital T is to capital T small t is to small t is mathematically condensable in the form of the binomial expression ax plus by the whole square that has the gametes bearing genes capital T or small t in equal frequency of 1 by 2, that is half. The expression is expanded as 1 by 2 capital T plus 1 by 2 small t, the whole square, is equal to 1 by 2 capital T plus 1 by 2 small t into 1 by 2 capital T plus 1 by 2 small t. That is equal to 1 by 4 capital T plus 1 by 2 capital T small t plus 1 by 4 small t. Though the genotype ratio can be calculated using mathematical probability, but it is not possible to know the genotypic composition by looking at the phenotype of a dominant trait. Therefore, to solve this problem, Mendel devised a test cross. A special type of back cross to the recessive parent is known as the test cross. Back cross is a cross of F1 progeny back to one of their parents. This method was devised by Mendel to determine whether the dominant phenotype is homozygous or heterozygous. Example, in a monohybrid cross between violet color flower W and white color flower small w, the F1 hybrid was a violet color flower. If all the F1 progeny are of violet color, then the dominant flower is homozygous, and if the progenies are in ratios 1:1, then the dominant flower is heterozygous. In case of a double heterozygous, that is heterozygous violet and axial flower crossed with double recessive, that is recessive white and terminal flower, the ratio will be 1:1:1:1. Here, another example is taken where capital T and small t is the hybrid tall of F1 generation plant and the recessive dwarf is small t. The gametes which are produced by this plant will be capital T and small t, whereas this dwarf plant will produce the gametes containing small t. Now when this gamete, capital T gamete, fuses with this small t gamete, it results in formation of capital T small t and the phenotype of that plant will be tall. Whereas if the gamete containing small t fuses with the gamete containing small t of the recessive dwarf, it will result in formation of a dwarf plant containing small t. Okay, so the phenotypic ratio is 1:1 and even the genotypic ratio is 1:1. Now, inheritance of one gene. Inheritance of one gene can be explained by monohybrid cross. The cross between the two parents differing in one pair of contrasting character is called monohybrid cross. Based on his observation on monohybrid crosses, Mendel proposed two general rules in order to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance, which we will be discussing after some time. But let us see about the monohybrid cross first. This is the monohybrid cross, where a plant containing capital T genotype, which is the tall plant, and another plant which is considered as the parent having the genotype small t is dwarf. When these two plants are crossed, the gametes which are produced by the tall plant will contain the capital T and the gametes which are produced by the dwarf plant will contain small t. Now when this gametes fuses, it results in formation of F1 generation plants which are all tall. But the genotype is capital T small t. Now when the self-pollination of F1 generation plants is done, the gametes which are produced will be capital T small t. Okay, so when self-pollination is done, it results in formation of 4 different plants where out of 4, the 3 plants will be tall, that is capital T small t and capital T small t. So 3 plants will be tall and 1 plant will be dwarf. Okay, here can you see the square? This is the Punnett square. So this was the square which was used by the Mendel for doing the crosses in his experiments. So this is Mendel's first law, wherein he said that homozygous dominant and homozygous recessive, when they are cross-pollinated in F1 generation, all the plants will be tall, which will be heterozygous, that is capital T small t. Here you can see they are homozygous. Same alleles are present. Here also same alleles. So all the plants of the F1 generation were tall. Now when the F1 generation plants were self-pollinated, okay, it produced 4 plants where 1 plant was tall. Sorry, out of the 4 plants, 3 plants were tall and 1 plant was dwarf. Out of 3, 1 tall plant contains the genotype capital T and the other 2 tall plants contain the genotype capital T small t, whereas the dwarf plant contain the genotype small t. Right, so if we consider the phenotypic ratio is 3:1. Now we have already discussed that phenotype means the observable features and the genotypic ratio is 1:2:1. where genotype is the genetic combination, that is capital T or small t or capital T small t together. This is the genotype. So here you can see there is one plant which has capital T. So this one for that. There are two plants which have capital T and small t. So 1 is to 2 is to 1, and the last one is for this small t. Okay. So I hope you have understood the monohybrid cross and the phenotypic and genotypic ratios of the monohybrid cross. Now, as we have discussed earlier, that Mendel proposed two general rules in order to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance, and they are law of dominance, law of segregation, okay, the first law is law of paired factors. Factors are responsible for transmission of characters, and these factors are present in pairs. Let us understand about the law of dominance. Characters are controlled by discrete units called the factors, and these factors occurs in pair. In a dissimilar pair of factors, one member of the pair is dominant while the other is recessive. This law is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 generation, and the expression of both in the F2 generation. It also explains the proportion of 3:1 ratio obtained in the F2 generation. Now, the second law is the law of segregation, which is also known as law of purity of gametes. This law states that though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Hence, the alleles do not show any blending and both the characters are recovered as such in the F2 generation, though one of this is not seen in the F1 generation. So that is about the laws given by Mendel. Now let us study about inheritance of two genes. To verify his results of monohybrid cross, Mendel also crossed pea plants differing in two characters, that is dihybrid cross. This helped him to understand inheritance of two genes, that is two pairs of alleles at a time. It was found that inheritance of one pair of allele, one trait, does not interfere in the inheritance of other pair of allele, second trait. Based upon this, Mendel proposed a second set of generalizations postulates which is called law of independent assortment. This law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters at the time of gamete formation. It also gets randomly rearranged in the offsprings, producing both parental and new combinations of parents. So this is the example of dihybrid cross. Okay, so dihybrid cross can be understood by this figure. where the parents are round and yellow seeds and wrinkle and green seeds. The genotype of round and yellow is capital R, small, capital Y, whereas for wrinkle green it is small r, small y. The gametes which are produced by this plant will contain capital R small y, whereas the gamete produced by this plant will contain small r small y. And when this is cross-pollinated, it produces the F1 generation, which will have capital R, capital Y small y genotype, and the phenotype will be round yellow. Now further, self-pollination of F1 generation plant is done. So capital R, capital Y small y is self-pollinated, and it produces different types of gametes like capital R, capital Y, capital R small r capital Y, and small r small y. Right, so when these are allowed to fuse with each other, when this gamete fuses with each other, it results in formation of 16 different types of seeds. Okay, so if we study the resulting phenotypes, we will find that the phenotypes are round yellow, round green, wrinkled yellow, wrinkled green, and these seeds are produced in the ratio of 9:3:3:1, whereas the genotypic ratio will be 1:2:2:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:4:44:1:1:2:1:2:1. This is what? This is genotypic ratio. If you will study this, the genotypes of different seeds, you will find that they are in the ratio of 1:2:2:4:1:1:2:1:2:1. And the phenotypes are in the ratio of 9:3:3:1. Now this is dihybrid test cross. F1 hybrid is crossed with recessive green wrinkled pea plant. This is F1. Okay, and it is crossed with the green recessive wrinkled plant. So the gametes which are produced will be, these are the gametes, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. And the recessive green wrinkled will have the gametes small r, small y. So when this are crossed, it results in formation of capital R, capital Y, small y. When this and this fuses, if this and this fuses, it results in formation of capital R, small y, small y. If this and this fuses, it produces small r, small y, capital Y. And if this and this fuses, it results in formation of small r, small y, small y. So the phenotypic ratio is 1:1:1:1. That is all for today. In our next video, we will continue the chapter in which we will be learning about deviations from Mendelism. I hope you have understood this. Thank you.